Thanks for inviting me here today. The first thing I'd like to point out is that I am a mechanical engineer by trade. So if any of you have any detailed questions on exhaust manifolds or cylinder heads, I'll be happy to answer them later. If I get any of the nomenclature wrong, I apologise in advance. So today I'm going to talk about the Living Heart Project. My talk really will follow on from where Praz was talking about before. The number of variables involved in an organ level model like this, the Living Heart, are huge, and the uncertainty qualification is very, very difficult. It's really important for us to be able to characterize and have confidence that the model performance is correct before we then go on to look at uncertainty qualification in the parameterization. So as an engineer, my eye is immediately drawn to the center image, which is the finite element model of the living heart. So what I'm going to do is introduce the heart model, the framework. I'm going to look at the physics and key attributes of the model, talk about the credibility assessment framework, and then look at some specific applications that we've used to quantify the model. So this is the model. It is multi-physics in terms of the fact that the muscle contraction is driven by an electrical excitation and produces a blood flow through the heart. So this is now running on our 3D Experience platform. I think it's the largest collaborative project of its type done anywhere to produce a commercial level model in such a way. So it involved clinicians, it involved academics, and it involved the FDA, the regulatory bodies, and some of the project members are in this room today. This is the same model rendered for some of our clinicians who prefer to see it like this. They're not being used to engineering type models with contours and what have you. This gives them a better idea of the motion. It helps them to understand better the motion rather than this sort of more engineering view that as an engineer you understand. So at the core of the Living Heart model and the Living Heart project and the framework, we've got modeling and simulation technologies. It's multi-physics and it's multi-scale. Then on top of that we can bolt extensions such as constitutive laws. So currently we use Alton-Holt-Sapfor and isotropic hyperelastic material models that also include the contractual effects, so it's active and passive. Then the models are built by our stakeholders, by our partners, and by us, by Dassault Systèmes as well. The models are accessible within the project, they're shareable within the project, and customizable, which means that we're trying to foster and accelerate the innovation in this. And this is very much a work in progress, and is sort of designed to show the way forward. I'm particularly, why I'm here today is because I think that this sort of life sciences technology within the bounds of what we know as engineering technology, proven technology, is really exciting and provides a really great future for humankind, and that's why I'm particularly interested in this. So we are, when we say multi-scale, what do we mean? Well, way from molecular interactions through biochemical pathways, cellular biophysics, all the way up to patient populations. Our heart model, our Living Heart model, fits here with tissue mechanics and the whole heart physiology. It's a phenomenological model, but it's extensible in both ways. We're now working with other softwares within our portfolio of products to look at molecular interactions and drug efficacy as well. So we can change the electrical stimulus to the heart model based on what's happening with the drugs, and using O'Hara-Rudy type models that have already been mentioned. But as has previously been said, that the runtimes on these are huge, and this is something that we're going to have to address in the future, particularly when you get to the whole heart or the whole organ level. And work that's being done at the University College London, looking at patient populations and how we tie that into the heart model. So what are the key attributes? It's a three-dimensional model of a healthy 4-chamber adult male human heart, taken from high resolution MRI scan. We use zygote media as well to characterize it into a CAD model that could be brought into an engineering finite element analysis code, which is Abacus. So Abacus has been used in aerospace and automotive since early 80s. It's proven technology within that industry. Part of the project was to see how this could be done, how it could be used in this sort of situation. So it's a sequential approach. As I said before, an electrical stimulus produces a mechanical response which drives a hemodynamic flow. So it's a hybrid 3D heart system model. We can look at disease cardiac function by modifying attributes, loads, boundary conditions, and material properties. The particularly good thing about it is medical devices can be added to study their performance. So we can look at not only at the effect of the device on the heart, but of the heart on the device as well, which I'll talk briefly about afterwards. So it is a sequentially coupled simulation workflow based on a 1 second beat. For those of you that are interested, my heartbeat is now 93 and it was 65 when I started, so I'm well above this. So some of the applications, we can look at device design, as I say, stent deployment, annuloplasty rings, that sort of thing, personalized models and disease. We're beginning to look at AI, machine learning, in terms of the blood model that's there, and also the drug efficacy type as well. With our electromagnetic portfolio now, we can look at virtual CT, MRI safety, what happens with devices in the heart when they're put in an MRI scan, and also personalized organs, 3D printing of organs and how they compare to the MRI scan that comes back, which is quite an interesting application. So moving on, V&V40, verification, validation. Is our model good? Do we know? How do we know it's good? It's a big model. It could be extremely useful, but obviously we need to have confidence in the performance. So all models, organ level, cell, subcellular, can be supported by any or all of the following types of credibility evidence, and this is something that perhaps has been published. So we need to quantify the model in terms of evidence relating to the validity of assumptions. As engineers, we make assumptions to get the analysis to run. That's what we do. We know that we can't represent it. It is an approximate method. What you have to do is to make sure your assumptions fit the context of use. Do we represent adequately what the end result? So we need evidence for the validity of assumptions, the accuracy or fidelity of the model parameter values, where they come from, why, how do we know they're good, calibration evidence, how do the results of our analysis tie, how do they line up with experimental in vivo measurements, how do they actually fit, and then validate that results and related evidence. And I think this is something that needs to be done before we actually go on to then look at the uncertainty quantification, certainly in our model of ionic channels or cellular level interactions. So, how we would see it, the green ticks mean we're kind of done. So, in view of our simulation results, as I say, Abacus is ISO 9001 tested. It's certified for aircraft and vehicle behavior. So, actually, the underlying technology is sound. Do we get agreement between expected behavior and metrics? Well, yes, I'm going to show that in a minute that we do. If we tune parameters, can we get the agreement? Yes. Do we agree with experiments where there's no specific context of use? Yes. But there's still work to be done in terms of specific context of use, and also the sort of parameterization of values in there. So this is where we stand with expected agreement. The simulation with valves compared to published normal ranges, you can see that generally we fit. the LV and RV pressures are slightly low, but it's work in progress. It's something that we need to be addressed, and all the time our partners and our R&D are looking at this to address these things. So going back to the categorization and looking now at category 2, which is, do the tune parameters produce agreement between the model and experiment? We were looking for published results for the activation times for the heart, and couldn't get them. So all we could do was base our results on specific positions within the model for activation time. And what we found was actually Dr. Serge Cazot came back to us and said, "I'm afraid your model is wrong. The activation times may be correct at the points that you've identified. However, the sequence is incorrect." So he was part of the project. We worked closely with him to identify what the problem was, and actually one of the branches of our Purkinje network, which we have included in the model, was not present in the model. So that meant that the activation process was wrong, and he helped us to correct that. We also looked at the activation physics actually within the code and changed that slightly as well, such that we got a better result for the activation. And this is our model on the right compared to patient data on the left. And you can see that we're a much better result. So category 3, general validation. This is work done at University College London by Claudio and the team. Qualitative comparison of cine images with comparable views. And in general we get the correct thickening, we get the correct rotation or torsion throughout the thing. So we throughout the process. So we can say that we do have a qualitative agreement with experiments for that context of use. A partner company or a part of the project, a team called Exponent, have done sequential studies year on year. In 2017 they compared the static and dynamic in vivo measurements of the living heart compared to in vivo. What they found was statically the living heart geometry represents average to above average heart. Dynamically the motions were in the 95th percentile. They were very close. And what they said at the time was that for device simulation, that we would represent a worst case scenario. So very good agreement. Then they moved on to look at coronary vessel motion. So we introduced into the model the coronary vessels on the outside, and they then went on to look at the comparison between in vivo and the living heart, and found that again were very representative, and we could use the model as a good representation for coronary artery stent deployment, etc. And then looking at pre and post device implantation as well. This study done in 2018 showed that we were again conservative loading environment, but justified to be able to look at device deployment or device simulation in these scenarios. So in category 3 of general validation studies, this was done by Kevin Sack when he was at South Africa. The study looked at 3 groups of pigs. So in addition to the living heart model, we have developed a porcine model as well. There's obviously a lot more in vivo data available for that. So this study looked at 3 groups of pigs. A healthy group, a group which had heart failure induced but not treated, and a group which had heart failure induced and then treated with hydrogel injections. Each pig heart was modelled individually, and looking at the strain results based on our model and the in vivo measured showed a really excellent agreement, I think, kind of proving that the methodologies can be used to create and simulate subject-specific hearts. It's more difficult from a fibre orientation point of view than you would imagine, but very accurate strains were predicted through the use of independent mechanical data. And models can provide insight into the treatment impact. The study went on actually to look at the effect on the local environment to the hydrogel implants as well, what was happening around there. But I don't show those results here. But it was a very good study done. And then just to move on to some medical device applications, a pacemaker lead, left ventricle assist device, TAVR deployment, leadless pacemaker, and looking at some undersized annually plastic devices. I'll fly through these because I'm conscious that we're running out of time. So this is particularly interesting. Before I started this job or got interested in this, I never realised that pacemaker leads don't come out. So it's important that they survive in terms of fatigue stress. So this was a study looking at where the fixation point should be, how much redundancy, that slack in the pacemaker should be. And this was done with the AAMI as part of the project, and it's published. The interesting thing is that it wasn't necessarily just the amount of slack that you would expect. It was a combination of factors. And using our eyesight software, we were able to sort of look at the combination of parameters which were more important, which is something that, again, that's been mentioned this morning. In fact, it's a combination of redundancy and ejection fraction, which are the significant factors in this. What this means is that when we're looking at clinical trials, that's where they should focus their attention. So this is where a use of where simulation is used to drive clinical trials. And I think as we come on to become, as the FDA gets more involved and their simulation working group approves more the use of simulation, and we get better in the work that we do in terms of confidence and uncertainty, then I think this will become more and more important, that simulation will eventually take over. And that's the aim of Dassault Systèmes, is to reduce animal testing and reduce clinical trials. So now I'm looking at the performance of left ventricle assist device. So this was done quite simply in our model by just reducing the contractility to achieve a lower ejection fraction, from a normal ejection fraction of 56% down to 28%, based on just changing or tuning the model to do that. Then running the LVAD with 2 speeds, based on the one-dimensional fluid flow model within the heart, to look at what ejection fractions could be achieved. The interesting thing is, if you look at the stresses at the end of diastole and end of systole, you can see that where it's green, they return back to normal after the event, but for the higher run speed, the right ventricle stress is significantly greater, doesn't return to normal. And in fact, what reported results shows that 10 to 30% of patients treated have right ventricle failure. Looking at leadless pacemaker anchoring in response, again, this is something that we can tune the model to do, based on a series of exercises and heart pressures and heart rates. Again, I'll be quick, but just in dark blue is the output from our living heart model, and in pale blue is the experimental output. We show excellent agreement based on Clint Hill, and this is where it actually goes. Of course, now once you're confident and you've got a base model as an engineering base, now we can look at different designs for what that anchor should be and how we can make it last in terms of life, based on a variety of load cases and boundary conditions. So we have a methodology for simulating elevated heart we can look at dynamic anchoring forces and then optimize a design based on those. This was a work done with Ellen Kuhl at Stanford. Again, our heart model, which we're confident in the results of, was dilated and grown, and then we looked at the effect of putting undersized annuloplastic devices in terms of the suture stretch and the local strain, myocardial strain. So just to show that we can look at the heart model provides us with a really good base for looking at these sorts of analyses. Again, this looked at structural simulation, optimization of deployment of a TAVR. The one we looked at in particular was the Medtronic Evolute R, and looking at also the effects of calcification on the leaflets were included. It was crimped and deployed at 3 locations: aortic, midway, and ventricular, to see which is the best way to go, which is the best way to model. We monitored the contact area and the contact force, and you can see that the aortic deployment is not a good one for that. Also within that, we can look at a hemodynamic response as well around the leaflet valves. It shows helical flow, it shows recirculation in the sinuses. And the takeaways from that. So the effect of the dynamic effect can have a significant influence on the anchoring performance of devices, etc. So just in summary, it's important, I think, to have confidence in the model that you set up. So we have, throughout the heart project, we've been looking constantly at making sure that our model, we feel, gives a good result or a good base, and from which further experimentation can take place. Now we've started within the project, we have 2 statistical groups. Beg your pardon, we have 1 statistical group, that's Xiaoyu Liu at Glasgow, and they're beginning to look now at the effects of uncertainty on various parameters within our model. There's nothing ready yet to be published. I hope there would be, but sadly there isn't just at the moment. And I've just recently been speaking with the University of Liverpool and their statistical group there as well. So I think, with a decent base model that we have here, the uncertainty qualification we can certainly take forward from a statistical environment. And with that, I would say thank you for your time, and I'll take any questions.
Thank you very much. Questions? Probably because of time, we may take 2 or 3 questions.
So for the coronary arteries, you said you represent this as, did you account for the heterogeneity of the coronary structure and also for the perfusion, like normal too?
Perfusion is something that we haven't looked at yet. Perfusion is not easy to model, and it's certainly in terms of a model of that size and complexity, I think it's not there. Really, the idea of including the coronary artery was to look at stent deployment and to give a more realistic load case for stents, so that you can quantify diameter, design, length, location in a more realistic environment in terms of the fatigue and life of the structure.
Any more questions? Can I just one question? So you were showing a number of different applications. It's very interesting, and one application you were showing is for the device, where you were looking at the geometry and the effect of the device on the geometry. Was you mentioned you extracted the mechanical information. Once that was studied, did you take it back to see whether the electrical signaling was affected in some way, and whether putting in that device could cause arrhythmia?
No, we haven't looked at that. This was purely mechanical. This was just looking at a mechanical response of the pacemaker lead in terms of fatigue, life again. That's what the study was for.
In general, is it something that more people want to look at that, or is it assumed that the electrical signaling just minimally affects it?
There is no reason why we couldn't look at that. The physics are all there. The location point, we can do that if that's what that's something that we could look at, a study that we could look at for sure.
Thank you very much.
Thank you.